All right, let me talk about the role of the antidiuretic hormone. This hormone has two names, used interchangeably, depends on the textbook that you have. Our book likes to call it vasopressin. Many other books call it the antidiuretic hormone. It is in fact the same hormone. So let's talk a little bit about ADH, the antidiuretic hormone, vasopressin. There's your brain with the pituitary extending down below the brain. And just above that pituitary is the region of the brain known as the hypothalamus. Neurons in the hypothalamus produce and secrete this peptide hormone, vasopressin. These neurons originate in the hypothalamus and extend down through the stalk of the pituitary, and the terminal region of these neurons release their peptide hormone into the bloodstream. These neurons in the hypothalamus that produce and release vasopressin are controlled by other neurons in that region known as osmoreceptors. And these osmoreceptors are sensitive to circulating sodium ion concentrations. When sodium ion concentration is elevated above set point, that means an individual's total solute concentration in their body has become elevated. When the sodium concentration rises above set point, those osmoreceptors are stimulated and in turn stimulate the neurons that produce and release antidiuretic hormone. If the total body osmolality, especially sodium, is lower than set point, those neurons and the osmoreceptors no longer stimulate the neurons that produce antidiuretic hormone. So let's summarize. If plasma osmolality is elevated, by elevated I mean above that regulated variable, that set point. If plasma osmolality is elevated above set point, we see increases in vasopressin, the antidiuretic hormone. Similarly, or I guess, alternatively, if that plasma osmolality is lower than set point, we see a reduction in vasopressin levels being released, a reduction in the antidiuretic hormone. So we have to appreciate the controlling factor for the antidiuretic hormone, for vasopressin. The controlling factor is plasma osmolality. Now let's go back to the nephron and see the role of vasopressin in the nephron. Now I've drawn only the collecting duct and the proximal tubule because we know that the filtrate entering the proximal tubule from the ascending limb in the loop of Henle is hypoosmotic relative to the rest of the body. That filtrate has a lesser solute concentration than the rest of the body. If the body has a total solute concentration of approximately 300 milliosmolar, a believable number for the filtrate that enters the distal tubule is approximately 100 milliosmolar, about 1/3 the solute concentration of the rest of the body. So let's draw 100 there. What's going to happen to this filtrate as it makes its way through the distal tubule and collecting duct? Well, that depends on whether or not vasopressin is available in any significant concentration. Now remember why vasopressin levels would be elevated. If a person's total solute concentration is elevated, so are vasopressin levels. So let's first assume this person's total solute concentration is above set point. They're a little bit dehydrated. This would result in increased vasopressin release from the posterior pituitary, and the vasopressin would act here at the distal tubule and the collecting duct. And what vasopressin does is it stimulates the production of water channels, aquaporins. So I've just schematically drawn two areas of the distal tubule and collecting duct. And vasopressin has arrived here because this individual's total solute concentration is above set point. Water channels are incorporated into these cells of the lumen of the tubule. And now, with these water channels, water will move out of the distal tubule and collecting duct to an area of high solute concentration. That area of high solute concentration is what the Lupa Henle has been developing in the renal interstitium. How much water will move out? The amount of water that gets reabsorbed here in the distal tubule and collecting duct depends on the amount of vasopressin and the extent of that interstitial osmotic gradient that the loops of Henle have developed. If the loops of Henle are producing their maximal interstitial gradient and the body is releasing its maximal amount of antidiuretic hormone, this final filtrate can be concentrated to the extent that the loops of Henle can develop the interstitial osmotic gradient. Remember what we drew before? The loop of Henle can develop an interstitial osmotic gradient that ranges from 300 milliosmolar to 1200 milliosmolar. So in the presence of maximal functional levels of antidiuretic hormone, this final filtrate can be concentrated up to 1200 milliosmolar. Theoretically. Exactly how concentrated does it have to be? Well, that depends on how much water needs to be reabsorbed to return the vasopressin levels back to normal. That is, to put an individual back in total solute concentration that's at their set point. If there's no antidiuretic hormone levels here, because a person, say, is a little bit overhydrated, in the absence of antidiuretic hormone, we get no further water reabsorption in the distal tubule and the collecting duct. And in this case, the final filtrate is the same solute concentration as the filtrate that enters the distal tubule, 100 milliosmolar in our example. So the human body is capable of producing urine that's very dilute, that's only 1/3 the solute concentration of the rest of the body, approximately 100 milliosmolar, all the way up to a urine that's 4 times as concentrated as the whole rest of the body, 1200 milliosmolar. So what is the urine osmolality at any given time? Again, the urinary osmolality is whatever level it needs to be to allow for water reabsorption out of the distal tubule and collecting duct in an effort to put this individual back into homeostasis with regard to their total solute concentration. High levels of antidiuretic hormone mean small urine volumes and a very concentrated urine with regard to solute. Low levels or no antidiuretic hormone means high urine volumes, which are very dilute, have a very low solute concentration. Let me summarize that on the board. Very high levels of antidiuretic hormone will produce a urine that has a very small volume and which has a very high solute concentration. On the other hand, very low levels of antidiuretic hormone or no antidiuretic hormone results in urine production very high in volume and very low in total solute concentration. The question to ask yourself is, why are ADH levels, why are vasopressin levels elevated above or below normal? Why is the release rate of vasopressin sometimes high? Why is it sometimes